This child is a victim of one of the very few inherited diseases that are understood in precise detail. New techniques of genetic engineering could save her life. But once we begin tampering with human genes, where do we draw the line?
There is the question or the concern that someday it might be possible to, in fact, manipulate our genetic composition and remake human beings.
As well as ethical, there are technical problems in applying this new science to human disease.
These kinds of manipulations still are problematical. They do damage to cells on occasion.
But some who are closely involved are impatient to begin this journey into the medical unknown.
Gene therapy is perhaps a little bit of a risk, but so is everything we try on these patients.
The time is now.
A parent, a child, and a physician willing to take the genetic gamble next on Nova.
Major funding for Nova is provided by this station and other public television stations nationwide. Additional funding was provided by the Johnson & Johnson family of companies, supplying healthcare products worldwide. And Allied Corporation, a world leader in advanced technology products for the aerospace, automotive, chemicals, and electronics industries.
A bug specialist for the butterflies in the stomach for $1,000.
That'll be a little easier.
6-year-old Erin McCarthy is not one of life's big winners. She lacks the natural immunity the rest of us depend on to combat the sources of infection all around us. Seeing her like this, it's hard to realize she has a life-threatening disease. That's partly because Erin, on her bad days, would be a very disturbing sight.
What? I don't like that.
It's also a testimony to the tireless efforts of her parents, her doctors, and expert home nurses.
You have to get me the rubber band.
Okay, Erin, let's
Okay,
get you
yeah, hook it up
started tonight,
first.
okay? Can we get you hooked
A
up for
bug,
tonight?
a bug, a bug.
Life in this house is dominated by the battle to keep Erin alive. Her playmates are discreetly screened. A minor cold could be lethal to her.
Do you want to empty these like you usually do?
Okay.
Erin, do you want to empty these like you usually do? Yeah, see?
The cause of her condition is the lack of a natural enzyme called ADA, a crucial chemical link in the immune system. That, in turn, was caused by an unlucky combination of genes coming together from both sides of the family.
Two more. That's fine.
But her parents had no idea this devastating legacy lay hidden within them.
No, we didn't. It had never shown up in a family history or anything. It came as a total surprise and shock.
The chances of one person who's a carrier marrying another person who's a carrier would be a quite extraordinary event. And that bears out in the clinical situation. We only see perhaps 30 to 50 new cases of ADA deficiency in the United States per year.
In diseases like this, simply injecting the missing enzyme does not help. It has to be manufactured inside the body cells. Since the key elements of the immune system are contained in the bone marrow, total bone marrow transplant is a possible treatment. But Erin has no matched sibling to act as a donor. Now, an experimental treatment called gene therapy is a scientific ray of hope for Erin. It would involve manipulating the cells of her own bone marrow, using the techniques of molecular biology. In short, bringing genetic engineering out of the research lab and into the hospital.
This is a fatal disease. Gene therapy is perhaps a little bit of a risk, but so is everything we try on these patients.
Medicine is a science, but it's also an art. And with every decision that we've ever made with Erin, we more or less compile the information, sift through it, and go with our gut feeling as far as if we really feel it's something that would benefit her.
It's almost a question of inevitability, unless you can, get something permanent, some permanent immunity. You can't walk around in white clothes for a year and not get them dirty. Well, that's similar to here. We've been very fortunate for 6 years, and many people that we've met and known haven't been so fortunate. And the time is now.
Inherited defects are responsible for about 3,000 diseases. Most are not exactly household names. There's no simple cure for any of these diseases, which account for over 25% of admissions to children's hospitals. The gene responsible for ADA deficiency is understood precisely. It's one of the 511 human genes on file at the National Genetic Databank.
That is nice because at a glance you really see the variation from one sequence to another.
Well, if we just take this oncogene sequence that we have here and display it.
These rows of A's, G's, C's, and T's are a gene, or the symbolic representation of one that's understood by geneticists and computers. A, G, C, and T are the initial letters of the 4 chemical bases that are used to form the links of the long chain which make up a real gene. It's a code, and in theory, if these scientists had enough of it in their computer, they would be looking at the specifications for a human individual, down to the tiniest detail.
One base position causes the glycine to change to a cysteine.
Biology, like everything else, has entered the age of the microprocessor. This amazing machine at Caltech can actually create a gene to order. The little bottles contain the chemicals needed to make the A, G, C, and T molecules. Punch in the required sequence, and the machine automatically assembles it. It's almost, not quite, making living things to order. This is just one dramatic example of the way biology has been transformed in a single generation. The science of observing nature is on the brink of becoming the technology of modifying nature when it goes wrong.
Yeah, she said she wanted.
So what's the connection between a gene and an enzyme like ADA, whose absence can have such devastating effects? These musicians are about to participate in a metaphor of genes and how they make enzymes and other proteins, which in turn make cells, which make animals like scientists, musicians, and the rest of us. Chords played by the orchestra will represent the component parts of big proteins like ADA. Notes played on the piano will represent the gene sequence. But it's a bizarre piano with only 4 notes, equivalent to the 4 genetic code bases A, G, C, and T.
We're ready to record.
2, 1, 2, 3.
Just as the composer can write any number of pieces with only 20 instruments, a biological cell using just 20 chemical components can assemble any of the 100,000 protein molecules that carry out the biochemical tasks of life. And a gene is a set of instructions for assembling a particular protein, decoded 3 units at a time. The combination GCT, for example, means go get a component called alanine and add it to what we're making. The musical chords on the lower staff represent the process of assembling a chain from those chemical components, the building blocks of life. The resulting chain detaches when the gene is completely decoded and wraps itself up to form the big protein molecule, which might be ADA, insulin, hemoglobin, or muscle. Superimposed on that process is the wonderfully complex hierarchy of cells, organs, animals. But the blueprint for the whole thing is in the genes, which are stored in packages called chromosomes.
The human chromosome, in a sense, plays the tune of life. There is a basic code that's present in the human chromosomes that consists of 3 billion different notes of 4 different types. If we divided these notes into bars and sheets of music, they would constitute a pile of music that's 2.5 miles tall. In humans, there may be 100,000 different genes, that is 100,000 different tunes. And as we go from a single egg, single-celled egg, to the adult organism with 1 billion different cells, in each of these different cell types, precise subsets of these tunes must be played to confer on those cells the characteristics of muscle cells or eye cells or red blood cells.
In actual fact, genetic information is stored in the chromosomes in 2 complementary copies, the famous double helix of DNA. So now the connection to genetic diseases can be understood. It's wrong notes in the music that cause them. At the Medical Center at the University of California, San Diego, dividing his time between clinical practice and lab research is Dr. Ted Friedman, who has long been attracted by the concept of curing genetic disease as simply as one might correct a misprint on a musical score.
Let's say this note is completely missing for some reason or other. That note has never found its way into this composition, or that base has never found its way into the gene. The consequence is to shift all the subsequent musical notation into new bars which didn't exist before, so that every bar is going to sound different.
Dr. Friedman is looking at a musical representation of a gene that specifies an enzyme called HPRT. Mistakes in this music cause a disorder at least as tragic as immune deficiency.
Wait a minute now. Let me do it this way, bro.
Called Lesch-Nyhan syndrome, the disease is a form of cerebral palsy, with a horrifying characteristic symptom of self-mutilation. Victims have to be restrained to prevent them from biting off their own fingertips.
You got another one? Oh, another one with a little more height? Huh? That's right, Mom. We're not worried about that right now.
Most of the 200 Lesch-Nyhan patients born every year have very short lives. Craig Weiner, thanks to the somewhat tough love of his brothers and constant home care, has made it to the age of 22.
You know what? I should go out and buy you some clothes today for your birthday. Right? How about a new TV? Yeah. Color? Color TV? Yeah. For the top of your bed? Yeah. Okay. With a remote control? Yeah. And a VCR? Yeah. For your tapes? You can tape the baseball games? Yeah. You can help yourself. Right, you can help yourself.
Once the disease was identified, UCSD researchers quickly tracked its cause. Amazingly, this complex syndrome is all due to the lack of that one substance, HPRT. When Ted Friedman isolated and described the HPRT gene in 1983, Lesch-Nyhan syndrome became a prime candidate for gene therapy. There is no conventional cure.
That's a quarter. That's a quarter. Okay, now I'm gonna move over here.
The reason for the pillows under his chin is to give him some sort of support because he has a tendency to throw his head forward, and it brought him a great deal of discomfort. And I thought it'd be best to have something to support his neck, and I couldn't think of anything else.
See, these kids come up with these things, one thing after another, like with the pillows. He would sit there and snap his neck, and snap his neck. You know, so all these different things used to come up. Like, he used to bite his lips when he was younger. But he doesn't do that anymore. He doesn't bite his fingers. He doesn't do any of that Mickey Mouse stuff anymore. Because he knows that it'll hurt him.
Yes, he's going to go to the airport today.
Friedman's lab team has been working quickly and carefully to show that they have a potential cure for Lesch-Nyhan syndrome. Working with cultures of bone marrow from Craig Weiner and other patients which carry the defective HPRT gene.
5%.
Is this Eric's?
Yeah, this is Eric's serum.
Hey guys, so how's it look?
It's looking pretty good. I'm getting some different morphology. I don't know if it's
These scientists have already developed a reliable way of inserting a good copy of the HPRT gene into cells in culture.
really different.
Since bone marrow is the factory for a whole complex suite of cells in the bloodstream and beyond, genetic transformation of marrow could have very far-reaching effects. That's been confirmed by animal experiments. But whether those effects reach the brain, and indeed whether that would be necessary to cure Lesch-Nyhan syndrome, is controversial.
Okay, here goes nothing.
That's lane 1?
Yeah, it's lane 1 and lane 2 from 2 different trials.
Another research team working on gene therapy at the National Institutes of Health is led by Dr. French Anderson. Anderson hopes to be the first scientist to bring gene therapy to humans, victims not of Lesch-Nyhan syndrome, but ADA deficiency.
Certainly, even after a week, there was enough human ADA activity.
To Anderson, ADA deficiency is the best bet because conventional bone marrow transplantation has a proven record in treating immune-deficient patients who have a matched donor. That cannot be said for Lesch-Nyhan syndrome.
Lesch-Nyhan has been the leading candidate for several years. Dr. Ted Friedman's work in San Diego has been pioneering in this. The difficulty is that the first attempt at a bone marrow transplant in a patient did not result in helping the patient. If you cannot help the patient with a full bone marrow transplant, it is unlikely that you're going to help them by correcting some of the cells.
Whether gene therapy has anything to offer patients like Craig Weiner is a dilemma for Ted Friedman. Scientifically, it's too soon to tell, but medically, there's a need to act before it's too late to help.
Okay, easy now. Okay, put your head back.
Many of us who see patients as well as try to study disease in the laboratory feel to be in a particularly difficult situation of having that clinical pressure, knowing what the clinical urgencies are, and treatment in medicine always involves a balance of known and unknown risks against the clinical urgencies to do something, even if one doesn't have perfect knowledge.
At the Whitehead Institute, affiliated with MIT, Richard Mulligan is a pure research scientist who is not personally subject to that clinical pressure.
The neatest thing is that down here it's a low fiber.
A leader in this field, he also considers ADA deficiency a better candidate for gene therapy than Lesch-Nyhan, but contends that it's too early for any clinical trials.
Suggesting that we go forward in the absence of this scientific information, I don't consider very scientific. I know the basic difficulty is you have three groups of people. You have the patients that are very, very sick. You have the physicians who want to help. And you have the scientists that will be responsible in a technical sense for the whole method. Well, I'm sure there's a lot of disagreement on this. My feeling is that one should not leave some of the scientific issues to the physician. Okay? We don't want to make decisions about the patient particularly, but especially in this case, I think I would certainly feel very, very more responsible than the physician for the patient if someone used a method I worked on.
For the last year, Mulligan in Boston, Friedman in San Diego, and French Anderson's group in Bethesda have been seen as competitive with each other and with a few other labs working in this highly advanced and fast-moving field of research.
And measuring the amount of activity in different clones. This was a positive control.
321?
14%. That's 14%. How do you like that?
Our whole group is now spending many, many hours every evening, all weekends, working in this. And it's really less the competition with other people as it is with nature. It's such an enormous question. The question of how can you get around nature's defense mechanisms and cure an incurable disease. It's very exciting.
Any plans for human gene therapy will have to be approved by this group, the Recombinant DNA Advisory Committee of the NIH.
Initially, what we've tried to do with our questions is to signal...
A working group on gene therapy has been debating the ethics of making genetic changes to sick children who cannot consent for themselves. French Anderson is himself on that subcommittee. Another key figure is an expert on medical law who was director of the President's Commission on Bioethics in 1982, Alexander Capron.
The notion that it is only for desperate diseases that desperate remedies are justified, actually I think can have a pernicious effect, because what it means sometimes is the notion that because of the desperation, you can try anything. And indeed, some of the recent heart experiments with artificial hearts and baboon hearts and so forth illustrate where I think that can sometimes go rather far awry, that notion that it's because we have nothing else to try, so we'll desperately try something. sometimes a careful examination of the risk-benefit ratio can go by the wayside, and also, particularly where children are involved, the notion that the parents may be put in a position of accepting something which might even in the end be harmful or cause a more painful death because there's nothing else that can be done. It is a very delicate issue. It's not to say that that isn't sometimes justified, but you have to be very concerned about the quality of the consent you're going to get.
The Weiner family's consent was underscored by their move from New York to San Diego to be ready at a moment's notice.
We all agreed.
I think it's worth the try, but what's most important, Craig thinks it's worth the try. Do you think it's worth the try, Craig?
You think so, Craig? Pick up your head.
Pick up your head, tell me. Tell me.
He said yes.
There's very few things that you do or that someone creates or someone invents or someone discovers that is undebatable. And simply because this issue can be debated, just like artificial hearts or anything else, to hold up the positive side of it, to me would be a very bad thing.
Comments from members of the committee?
To some, the scrupulous federal committee procedures have been taking precious time. In April 1985, the working group published draft guidelines for human gene therapy and invited public comment.
We very much appreciate the letters which we received.
One of those letters was from Aaron McCarthy's doctor, Sheldon Horowitz, reminding the subcommittee that lethal diseases work to their own, more urgent timetable. Gene therapy may have only a small chance of success, he wrote, but its risks are minimal compared with certain death.
We can't wait forever. We have gotten into trouble hearing that gene therapy might be available in 6-12 months and trying to wait, and while we wait, the kid gets some terrible infection, and then we're really up against the wall.
French Anderson was one of many who took notice. He contacted Horowitz, and the two of them began drafting the paperwork they'll need for permission once they're technically ready.
If we run into a major problem, and other laboratories, other groups working on this run into major problems, it could be a year, 2 years, 5 years, it could be never. But at present, we anticipate that probably sometime early in 1986.
So it seems that some very sick child will soon begin a journey into the medical unknown. Since gene therapy has not yet been tried, the patient and her mother in this scene are portrayed by actresses. The Hutchinson Cancer Center in Seattle is a leading bone marrow transplant center for leukemia, which may become involved in gene therapy. Its medical and research staff cooperated in this simulation by playing out their own roles in the procedure.
And just after you taste that funny taste, you'll fall asleep. And when you wake up, we'll be all done. Okay? Okay. Good night.
How is your blood pressure?
Withdrawing a sample of bone marrow is not a major or very lengthy procedure, but the surgeons need to dig deep into the hip bones and withdraw about a pint of marrow to be sure of harvesting enough of the actively dividing cells, whose offshoots will spread throughout the body. In the biochemical lab, the marrow will be selectively cultured to favor those special cells. They're called stem cells. But since stem cells can't be isolated or even identified, it's impossible to be sure how many of them will absorb the new gene in an effective way. And nobody will really know how many is enough until they try it on the first human patient.
How does the cell count look?
I'm just checking it now.
The gene is not simply mixed with the cultured cells, it has to be targeted to the cell nuclei. To achieve that, scientists use a naturally occurring organism called a retrovirus, cleverly adapted for genetic engineering. It takes an electron microscope to actually see a retrovirus. These tiny, primitive life forms reproduce by invading animal cells, often causing tumors. A retroviral gene, here symbolized by 4 bars of music, is actually many hundred bars long. And something in its special top and tail gives it the amazing ability to insinuate its own genes into those of an animal cell, forcing the cell to play the virus's music as well as its own. For use in gene therapy, scientists delete the virus's own harmful genes and substitute the gene for ADA or HPRT. The retrovirus goes about its work unaware that the music the cell will now play will be a passage from some other concerto altogether.
Get up onto the cart, get up onto
The next step for the patient will be a dose of gamma radiation that would normally be lethal. The point of this drastic step is to kill all the bone marrow cells remaining in the body, so that the genetically corrected cells, when reimplanted, will not compete for survival with a much larger number of unchanged cells. Patients with ADA deficiency will be spared this trauma because their already damaged immune cells have little power to compete.
Love ya. Love you too. Okay. Bye, Joni. Bye. See you in a bit.
But treatment of other genetic diseases will involve radiation. It's the point of no return. She must now get her bone marrow back or die. The marrow, bearing its new genetic information, does not need to be reimplanted surgically inside the bones. It will infuse gently and easily by IV drip. But this will be a crucial time for the patient, vulnerable to the slightest passing infection. There are many other technical problems to be overcome before gene therapy has any guarantee of success. One potentially serious one is that the retrovirus breaks the music of the host cell and inserts its own at a totally unpredictable point. This capricious behavior is baffling even to experts like Dr. Inder Verma.
We have absolutely no control on it. In fact, that's one of the weaknesses of all the work done on mammalian systems now, is that we don't know where genes integrate when you put them in a cell.
That means that the control of that gene is likely to be not quite normal. It also means that some of these events will result in the turning off of otherwise normal and needed gene functions. So these kinds of manipulations still are problematical. They do damage to cells on occasion.
Furthermore, there's also the problem that since these retroviruses have such strong elements of control for activation of RNA, for example, that there's no guarantee that they may also integrate in the vicinity of a gene which has the potential to become a tumor gene.
If genetic disease is a sour note in the score of a symphony, gene therapy is the equivalent of inserting a corrected page in the score somewhere at random. Whether the whole piece will then play well is truly a gamble.
Looks like you're ahead of me.
Our simulated child patient will be gambling with many ifs beyond the considerable risks of the bone marrow transplant itself.
How would you describe yourself to someone who does not know you?
If the gene got into enough stem cells, and if it did not disrupt another gene, and if the inserted gene makes enough of whatever enzyme she lacked before, then she will get better.
That sound good?
Yes.
Yeah? Okay, it's your turn.
I don't feel like playing anymore.
And those are the questions that will hang over the first gene therapy trials. The real patient is certain to be a child with one of the handful of rare diseases whose genetic basis is understood. The vast majority of diseases on this list are said to be genetic only because they're observed to run in families. The missing substance or the defective gene are unknown.
It is perhaps a paradox that the initial candidate for gene therapy is such a rare disease. It could easily have been a much more common disease, but we have so little knowledge about most human genetics that we really need to have a great deal more information about human genetics in general before it will be possible to apply these sophisticated techniques like gene therapy to a broad range of very common genetic diseases.
Further progress depends on unraveling more of the mystery locked up in the double helix. This is DNA itself, spilled from the single chromosome of a bacterium. Human DNA is packaged in 23 pairs of chromosomes, each of which may carry some 10,000 genes. The detective work involved in tracking down a gene to a precise location in all this is complex and fascinating. Since chromosomes are a family affair, the starting point of the detective work is to go out and talk to families with a genetic defect.
Daughter?
Yes. Her parents are both living. Okay.
These researchers are from the Howard Hughes Medical Institute in Salt Lake City. Since they give an absolute guarantee of anonymity to the families they work with, this scene is a simulation using a volunteer family that is not part of the study.
Now, I understand that Stephen has 7 children?
7. No, 8.
Oh, so
We
if
left Joshua out. Where does Joshua go? He goes between the Enoch and Nakai.
Once the family tree has been sorted out, it's time for some bloodletting.
While it may look like we have a lot of tubes here, we only collect about maybe 1/4 of a cup.
I'll go. I want to do it.
He doesn't need anybody else.
The right arm or the left?
We'll look at your left arm, okay?
I've got a wound in my left arm.
Oh, well, for heaven's sakes.
I just got that today from the rose bush.
There we go.
The white blood cells, which contain the chromosomes, are separated out and added to their cell library, a miniature high-rise city of 10,000 citizens in deep freeze. If you're looking for a gene and you don't know what it is or where it is, the only possible approach is to sample a large family and see what differences you can find between the chromosomes of those that have the disease and those that don't. In fact, it is now known that up to 90% of the music of the gene is not decoded bar by bar, but is unstructured and unannotated. By comparing the different way family members play those long passages of apparent nonsense, researchers get important clues as to where disease genes may lie. Chopping up chromosomes and decoding the pieces, they hope to find one particular pattern somewhere in the nonsense music that always crops up in the chromosomes of disease victims.
And then next, elastase.
If healthy family members always have a different pattern at that position on the chromosome, then that position is said to be a marker for the disease. Beginning with a general search of all 23 chromosomes, computer analysis eventually finds one chromosome for which this relationship almost always holds good.
1328. No, they're dead. Oh, okay. They're all dead.
Let's say a large family has 3 different marker positions on chromosome number 11. Call them A, B, and C. A form of diabetes also runs in this family. If everyone who has the diabetes also has marker pattern B, and nobody who is healthy has B, that's evidence that the diabetes gene is on chromosome 11. But that's only narrowed down the search to one chromosome, which carries perhaps 10,000 genes. Pinpointing the gene to an exact place depends on a curious feature of the way chromosomes are inherited. Here's an imaginary family of 2 grandparents, 4 children, and 3 grandchildren. And here's the biology textbook version of how any pair of chromosomes is handed on, starting with grandfather and grandmother. Each of their children inherits one from each of them, chosen at random. So let's say the eldest daughter gets a pink from father and a green from mother. The next born son gets a yellow and purple, and so on down the generations. Well, it's not quite that simple. In the formation of the all-important sex cells, the sperm or egg of any individual, the chromosomes actually merge in a complicated double X formation. When they separate, the X's are found to have exchanged parts in some random way. So what a son inherits would almost never be pure pink, but a mostly pink chromosome with some stripes of yellow from his other parent. The next generation gets a chromosome salad of all the family DNA. These crossovers, as they are called, have profound consequences for the inheritance of those markers that geneticists follow. If we represent a bad gene by a black square and a marker by an arrow, it's obvious that unless they're extremely close together, they risk being separated by crossovers. The number of times a marker and a gene wind up on different chromosomes is a measure of how far apart they are, so geneticists turn these random crossover events to their advantage, patiently counting how many members of a large family get the gene but not the marker, or vice versa, and deducing exactly where the gene must be.
I guess it wouldn't matter, because with the recombinant, their kids would just take the genes as they come recombinant with the recombinant in it.
Nowhere in the world is there a better example of that process than the shores of Lake Maracaibo in Venezuela. In the spring of every year since 1979, an American-led and funded expedition has set off for a remote corner of this huge inland sea 10 degrees north of the equator. The expedition leader is Dr. Nancy Wexler, president of the Hereditary Disease Foundation, which was endowed in memory of her mother, who died of Huntington's disease. The gene Dr. Wexler is tracking down with such passion is the gene that killed her mother and causes the lingering and undignified death of 1,000 other Americans a year. Their destination is a small village perched on stilts over a shallow lagoon. The traditional life of this community has been modified by a few gifts from the high-tech world elsewhere, notably the Japanese-built outboard motor. But there is no fast food, no air conditioning, and no medical care whatsoever. The diet, the culture, and the way of life revolve around one commodity: fish. Another fact of life here is the scourge of Huntington's disease. A single woman brought it to this area in the 1880s. The gene is dominant. Inheriting it from only one parent is sufficient to give the disease. And the random shuffling of the chromosomes over the generations since has distributed the disease to some 1,000 people in this region. Nobody in this village is without many relatives who suffer from it. And because the symptoms rarely appear until quite late in life, these people are condemned to live 40 years in the knowledge that genetic fate may have dealt them a very bad hand. And as the 40-year-old child of a victim herself, Nancy Wexler understands that predicament from a very personal point of view.
Each child of a parent with a disorder has a 50-50 risk of inheriting the disease. So I have a 50-50 risk of inheriting it. My sister has her own 50-50 risk of inheriting it. And there is no way at the moment to tell, there has been no way to tell who is carrying the lethal gene until you actually develop the symptoms of the disease. It causes uncontrollable movements in all parts of the body. It causes massive personality changes, often severe depression, suicidal depression, sometimes hallucinations, delusions. It causes cognitive decline, but patients have still very good insight into the fact that they are slowly and progressively losing all of their faculties. It's invariably fatal, and as of yet, there's no effective treatment.
Every day while she's here, Wexler sets off in search of missing links in the vast family tree that she and her colleagues have compiled over 6 years. The interlinked families of this region are a research goldmine. Thanks to data collected here, the Huntington's disease gene has already been linked to a marker on chromosome number 4 that inherits with the gene 95% of the time. And this has already given a way of predicting, for this family, who has the bad gene. Nancy Wexler explains how, using another imaginary family tree.
Imagine that these dominoes here are people. The black ones have Huntington's disease. And in fact, this one has died of this disease. The white ones here are unaffected. And these are 3 children who want to know what their fate will be. Each one of these children has 2 chromosome 4s, one from the mother, one from the father. And they have a different DNA pattern on each chromosome 4. In this case, you don't know which pattern came from which parent. You can see that these 2 children have the same patterns. This one has a little different one. The question for these children is, each of them has a 50-50 risk, which is going to get the disease. If you have the parents, you can see what patterns they have. This healthy mother has a 4-point pattern on each of her chromosome 4s. So all she can do is pass on the 4-point pattern to her children, which she has done right here. The question is, which of these 2 patterns in the sick father is sitting next to his Huntington's disease gene? You need to look at another generation. Luckily, this man's mother is still alive. His father has died of the disease, so his genetic information is unavailable to us. The mother has a 2-point pattern on each of her chromosome 4s. So the 2-point pattern has to be the healthy pattern, because she didn't have the Huntington's gene. So the 2-point pattern went to him. This was his healthy chromosome 4, and he gave his healthy chromosome 4 to these 2 children, so in fact these 2 children are free. The 5-point pattern has to be sitting next to the Huntington's disease gene. So in giving his 5-point pattern to this child here, this child will develop Huntington's disease.
Mire a las líneas. Mire a las líneas. Verde.
It takes no great expertise to see that Carmen Soto is a black domino. The neurological tests are not for diagnosis, but to record the progress of the disease since last year. Carmen is a direct descendant of the original carrier, and she married another Huntington's victim. So her offspring, with 2 black dominoes stacked against them, have a 75% chance of getting the disease.
Puede. Algo como yo. Por favor. No tiene cédula. No, tampoco tiene cédula.
Dora is one of Carmen's daughters. She's at the age when every time your eyelid twitches or you drop something, you worry that it's the onset of what they call here el mal. The blood and skin samples she is now consenting to give will tell an expert half a world away what her fate is. But Dora will not get the news. Even if she tests positive for the bad gene, that would not be a diagnosis. There's a 5% chance of error, and there's no treatment to offer, especially in this remote region. It's a major ethical problem which needs to be resolved, since many experts believe predictive tests will have at least as much impact on medicine as gene therapy. In this case, the researchers have decided for the time being to keep the information contained in these gifts of skin and blood to themselves. In a race against deterioration from the tropical heat, the samples are flown to the lab of Dr. James Gazella at Massachusetts General Hospital. Technicians immediately begin the process of chemically chopping up the chromosomes, looking for a closer marker. Somewhere among these fragments of Venezuelan DNA is their ultimate goal, the gene itself. And that could be found in 2 ways. Either find out what it does or how mistakes in it cause the disease, or track it down so tightly with these methods that you can find it and decode it. Either way, Dr. Gazella cannot promise gene therapy for Huntington's disease.
It's extremely hard to predict whether it's a candidate or not because it depends totally on what the defect is. If the defect is the production of something that is toxic, it's very unlikely that putting in a good copy of the gene is going to eliminate that production. Other techniques, which also involve gene therapy, are, however, being developed to eliminate activities of genes that are present, and those could then be used. If, however, the defect is the absence of production of some protein, then it is a potential candidate for gene therapy.
Meanwhile, does the success of this project offer any hope to Nancy Wexler personally?
Absolutely, but not immediately. In order to look at a family and see whether or not a person has the marker which says that the gene is there, you have to have enough of these relatives to look at. And unfortunately, in my family, everyone was wiped out by the disease. So for me personally, it will take the actual discovery of the gene itself to be able to do diagnosis for me and for my sister. But there are ways that we can now tell if a fetus is carrying the chromosome 4, say from my mother, in which case the fetus would have a 50-50 risk like I do, or from my father, in which case the fetus has no risk because he's not carrying the gene. So it's possible for me and for other people who don't have sufficient family, it is possible to give us some information about the health of future generations.
In the future of genetic research lies the possibility of not merely predicting, but actually controlling the health of future generations. When more of these diseases are genetically unraveled, a corrective gene could be inserted not just into general body cells, but into the sperm or egg, the germline. That gene would then be passed on to the patient's descendants forever. Bacterial gene factories in Dr. Leroy Hood's lab are producing millions of copies of the gene for a protein called myelin basic, as part of a futuristic experiment in germline engineering. The gene for myelin is defective in a mutant strain of lab mice called shiver mice. They shiver because they have no myelin to insulate their nerve fibers. Under a dissecting microscope, the uterus removed from a recently fertilized female shiver mouse is carefully torn to release the fertile mouse eggs, bathed in a milky fluid. One nucleus of each egg is going to be injected with a myelin gene, and every single cell in a mouse that develops from it, including its sex cells, will contain the foreign gene. So the change will be permanent and inheritable. The myelin gene, wrapped in a virus-like carrier, is drawn up into a hollow glass needle, the finest that can possibly be made. This experiment is futuristic not only in its implications, but in terms of sheer technique. The target, the upper nucleus, is less than 10,000ths of a millimeter across. When everything's in place, the gene is injected. Hundreds of copies whose exact destination in the chromosomes is unknown. A short time later, the eggs are inserted into the uterus of the anesthetized host female to develop normally. One of the most interesting results of this experiment was that early attempts failed. There was no doubt that the myelin gene did get inserted in the chromosomes, but that was not good enough. The gene was not turned on, and so did not make myelin. Within those billions of notes of the genetic concerto that are apparent nonsense, there are themes that have the vital role of controlling the genes that do code for proteins.
Indeed, we've begun to learn that there are important motifs in and around the gene that tell a gene how fast to work, tell the gene where to work, what particular cell type to be expressed in, and when to work in some cases.
Richard Palmater, working with Dr. Ralph Brinster of the University of Pennsylvania, provided the world with a dramatic demonstration of these control motifs in 1982, when they inserted growth hormone genes into mouse eggs. Attached to the growth hormone gene was a powerful control sequence from a different gene, one that responds to trace metals in the diet. Sure enough, when given zinc, the mice grew to giant size, and the experiment made scientific headlines. This experiment was sensational for what it taught us about gene control. but it does not necessarily herald the age in which inheritable changes can be made to human germ cells. The technique is not yet reliable, even in animals. So, from the ethical perspective, Alex Capron would support a ban on human germline therapy in the present state of knowledge, but not forever.
Reasons to be concerned about it. Beyond that, however, I think some of the notion that there should be an absolute prohibition on germline therapy is very puzzling. If there were certain conditions, and I think that there are, Leishmanian might be an example, which apparently have no benefit for the individuals, in either the affected individuals or the carriers of the disease, and which are devastating, and which may be complex to intervene with and so forth. Why not want to eliminate them not only in the patient, but in the patient's children and grandchildren and so forth?
Dr. Hood, who directed the Shivermouse experiment, is the first to agree that social dilemmas raised by the kind of work he's doing cannot be resolved by scientists alone.
I think all of us have opinions, and I don't think any of us necessarily is going to be right in our opinions, and it's my strong feeling that one is going to have to get together a panel of different people with different interests and different expertises to consider these questions in detail. Myself, as an experimental biologist, I think these kinds of opportunities afford absolutely mind-staggering possibilities for the future. And because of the exponential growth and the potential and the way in which we can carry out molecular biology, I suspect in the very near future we're even going to have more staggering opportunities.
This document is
A model for an ethical panel already exists, the working group of the NIH Recombinant DNA Advisory Committee. These meetings are open to the public, and members have been made forcefully aware of the deep fears that human genetic engineering can stir up. Jeremy Rifkin is an author and advocate of extreme caution. Fearful that decisions will be made by a scientific elite, he has taken full advantage of the committee's open policy.
A wide range of people. This is not just a medical issue. Human genetic therapy is a social issue. It's a cultural issue. It has long-term repercussions for the future of the human race. And even though we're at a very initial stage, the precedents we set over the next 5 years in this country will be looked to over the next 25 years by other countries.
Indeed, what may become possible in 25 years is very much on people's minds here, as they debate the ethics of using gene therapy on hopelessly sick children.
There is the additional ethical question of using therapeutic techniques for simply enhancing characteristics, for people to try to be more intelligent or bigger or better looking or whatever. The technical ability to do that really isn't present at the present time, because so many of the major characteristics like personality and character and so on are dictated by hundreds of genes interacting in totally unknown ways, interactions with the environment and so on. But there is the question or the concern that someday it might be possible to, in fact, manipulate our genetic composition and remake human beings.
Every time there is a potential to introduce a gene, there is no saying that what kind of gene can be introduced. Right now we are talking today of the genes which have a human potential of disease therapy because they're deficient, but I see it's perfectly possible somebody might want to introduce in the same way a gene which has a totally different motive behind it. So I personally think the public has a reasonable fear for it, and I think it is our job, the scientists, to explain to them that there are benefits of this one, and that there are safeguards which at least morally we try to apply. We can't really force on everybody.
I can't say that this kind of technology is going to be safe from abuse. On the other hand, who of us can really look at a child with Leishmanian disease or ADA deficiency or cystic fibrosis for that matter, and feel that that's normal and that that's not screaming out for correction?
I think it really becomes very questionable whether it's right for third parties to come in and say, we have some generalized concerns that in 20 years this may have some bad effects because we may learn something which could be misapplied by some malevolent scientist someday. Therefore, your child should not get a treatment which you and a review committee and an NIH committee have all said is an appropriate step to try to cure that child.
anything in this world that's abused can have negative consequences. The issue that we're dealing with from our perspective is that this could save lives and create futures.
The dilemma is, in taking this first step along the road to control of our genetic destiny, we would be creating futures not just for Aaron McCarthy, but for mankind in general. Do we have the wisdom to take that step?
For a transcript of this program, send $4 to Nova, Box 322, Boston, Massachusetts 02134. Please be sure to include the show title. Major funding for Nova is provided by this station and other public television stations nationwide. Additional funding was provided by Allied Corporation, a world leader in advanced technology products for the aerospace, automotive, chemicals, and electronics industries. And the Johnson & Johnson family of companies, supplying healthcare products worldwide. To purchase film or video copies of this program for educational use, call toll-free 1-800-621-2131. In Illinois or Alaska, call COLLECT 312-940-1260.